Alright, good afternoon, students. Alright, welcome to today's practical. Today we are looking at biology practical, experiment 5 today. The title of the experiment is Microscopic Examination of Bacterial Motility. The aim is to determine the motility of the bacteria using what? A microscope. Now, before we go on, what do you understand by motility of bacteria? When we talk about motility of the bacteria, we're talking about the ability of bacteria to want to move. Movement of what? Bacteria using what called energies. These energies are called metabolic what? Energies. Bacteria have what? Energies, just like what human beings want to have what? Energies. They are also living what? Things, because they are able to carry out what? Some characteristics which living things also what? Carry out. Talking about the plant and what? The animal what? Groups. Are you following? Now, the apparatus we are using today materials are what? Microscope, microscopic slide, copper slip, pond water, petroleum jelly or Vaseline, cotton wool, ethanol, pastel pipettes. Then the next thing is what? Is the procedure. The procedure is the method we use what? In arriving at observation and conclusion. Now the procedure involves sterilize the microscopic slide with ethanol using a sterile cotton wool. Place a drop of the cold water using a pastel pipette. Form a right angle using the cover slip. Seal the edges with the aid of the petroleum jelly Vaseline in order to form a cavity. Place the wet mount on the stage of the microscope and view. Now, since you are using cold water is what? Is a wet mount. Now, there are some techniques we use what? In carrying out this wet mount techniques. It's either we use what? The hanging drop techniques. And when we are using the hanging drop techniques, we are expected to use what? A cavity slide. But since we don't have a cavity slide, we are going to use the petroleum jelly to form what? A cavity. So that we can place the water and the water will not dish out. Are you following? So that's what we are going to do. We are going to use the petroleum jelly to form a cavity. And we enable us what? Hold the water so that we can be able to view what? The organism. Now, the next thing is what? Is the experimental setup. Experimental setup is a microscope having the microscopic slide as well as the petroleum jelly together with cover slip. Is your what? Experimental setup, because we're making your what? The microscope. Now the next thing is your what? Observation. When you're carrying out the motility test of bacteria using a wet mount techniques, you expected what? To view bacteria. Now bacteria such as Paramecium, amoeba, you see them as well as what? The algae, the green algae, or what? Or the mold. You see them on your what? On the microscopic slide using the 4 times objective lens. Now when you see that becomes what? Your observation. Whatever thing you have seen on the microscopic slide, you take a detailed diagram of that together with the level. Now because of what? Your observation. I have told you, you see organisms like the Paramecium, you see amoeba, you see what? The green algae, you see also the molds on what? The slide of what? Of the microscope. When you view using what? The 4 times. Now the next one is what? Is precaution. I've told you before, precautions are the safety measure what you carry out in order to what? Ensure what? A better what? Result. And with me then, finally conclusion. I've told you, you complete your practicals in respect to the what? To the aim. And the aim is to determine the motility of what? Of the bacteria. Using what? A microscope. Now what will your conclusion look like? You say that at the end of the experiment or the practical, bacterial motility was determined using what? The microscope, because the conclusion is very what? Simple. Are you following? Yeah. That's what we are going to what? To carry out now. So I'll go to the workbench and I will do what? The wet mounting with what? The aid of what? The microscope, so we can view these bacteria. But in order to what? To notice the motility of what? The bacteria. So let's go to the workbench and see what we have. Microscope is already on. This is the light source and this is the microscopic slide. These are our pond water from 2 different sources. This is our Vaseline, this is our beaker, and this is our pastel pipettes that will enable us to take the sample. Making your first water sample here. Now, this is the Vaseline or the petroleum jelly. I'm going to form a cavity. Now I've done two, I'm going to do two rather. I'm going to do the hanging drop and then the cover slip method. What I'm doing now is the hanging drop techniques. Now with the aid of the pastel pipette, I want to introduce the water, the pond water that we're going to view. This is the first sample of the pond water we're going to do for the two. Now I've done that. I'm going to mount it on the microscope. And I'm going to be using the 4 times objective lens, which is the smaller one. Okay, I've done my own using the hanging drop techniques. I can see the green algae and I can see some mortal bacteria on the stage of the microscope. So you're going to on your own light source now and carry out the same procedure, and I'm coming to inspect each and every one of you. So these are the materials. You can come to pick them. You can come to pick them. If you are planted your own, don't step on it. Let me continue. All right, let me see.
Precious.
Yes, have you tried it? All right,
Come on.